Hi all, welcome back to our Allied Lecdem channel. In this video, we are gonna see about what is blood group and some types of blood group like ABO blood groups, RH factors and importance of knowing blood group. So let's get into the video. First of all, we'll just recall what is blood. Blood is a specialized connective tissue consisting of a fluid matrix plasma and a cellular portion called formed elements. So, it is usually made up of the combinations of red and white blood cells and platelets suspended in a fluid known as plasma, that is, the liquid form of blood. And there is a million of molecules with their specific roles and functionalities. The composition usually varies from one human to another. Now we'll see what is blood grouping. Blood grouping is also known as blood typing. It is a classification of blood based upon the presence or absence of antigens and antibody substances on the surface of RBCs. So, these antigens may be proteins, carbohydrates, glycoproteins, or glycolipids depending on the blood grouping system. So, we'll just recall the history how this blood grouping system has been found. When blood from two individuals is being mixed, that is, sometimes the doctors had seen clumping of RBC occurs, that is, agglutination of RBC. Agglutination is because of the immunological reaction, but why it occurs in some cases and not in other cases created a mystery until the discovery of the blood group by the great Austrian scientist Karl Landsteiner in the year 1901, along with his co-worker, recognized that there are four types of blood groups in human beings commonly known as ABO blood grouping. But like this way, how the blood grouping system has come into an existence. Now we'll see the types of blood groups. Typically, the blood components are same in all humans, but there are different type of blood. In particularly, there are more than 40 blood groups, but not all of them are clinically significant. In this video, we are going to concentrate about two major important blood grouping system. They are ABO blood group system and RH blood group system. First, we'll see about ABO blood group. Before entering into the blood grouping of ABO, we should know what is antigen and antibodies, because these are the sole characteristics found in the blood, so which is main important for classification the blood grouping. So, antigen refers to a protein molecule present on the RBC surface, whereas antibodies are the proteins present in plasma. These elements of a natural defense system in the human body, when there are foreign substances like germs, antibody typically alert the immune system to destroy them. So, Landsteiner found there are two antigens on surface of RBC and named them as A antigen and B antigen. These antigens are also called agglutinogens. In simple, antigens are the protein which is seen in the RBC surface. And why they are named as agglutinogens? Because they have a capacity to cause agglutination, that is, clumping of RBC. And he also noticed a corresponding antibodies, which is also known as agglutinin in the plasma, and named them as anti-A or anti-B. A particular agglutinogen and the corresponding agglutin cannot be present together. That is, a particular agglutinogen, that is A antigen and anti-A, cannot be present together in a person. So, Landsteiner formulated a law stating if a particular agglutinogen, that is antigen, is present in the RBC, corresponding agglutinin, that is antibody, must be absent in the serum. Or else, if a particular agglutinogen is absent in the RBC, the corresponding agglutinin must be present in the serum. So, the antibody present together with antigens are found as follow. That is, antigen A with antibody B, Antigen B with antibody A, antigen AB has no antibodies, whether antigen is nil, that is a group O with antibody A and B. By knowing this law, we'll be able to classify the ABO blood group system. First, we'll see group A, that is blood group A. People with blood group A contain A antigen on the surface of RBC, so there is an anti-B antibody in the plasma. Anti-B antibody typically tends to attack the blood cell containing B antigen. So, why it attacks? Because if it comes, there will be a agglutination occurs in the blood which may result in hemolysis. So, people with blood group A can safely give blood to others matching with A blood group and AB blood group. However, they can only take the blood group from A and O. Why they could take a blood from O? Because they don't have any antigens. Next is group B. People with blood group B contain B antigen on RBC and anti-A antibody in the plasma. Anti-A antibody typically attack the blood cells containing A antigen. With people with the blood group B can safely give blood to others matching with B and AB blood groups. However, they can only take blood of group B and O. And the explanation goes as for the group A. Next is group AB. So, the blood group of AB contain both the antigens such as antigen A and antigen B on the surface of the RBC. So, since they have both antigens, the antibodies they doesn't have in their plasma. So, it does not have anti-A or anti-B antibodies in the plasma. People with blood group AB can safely give blood to those who have the presence of type AB blood group. Besides, they can take the blood of any blood group type. So, next, we'll see about group O. The blood group O does not contain a antigen or B antigen on the surface of RBCs. However, it includes both the antibodies such as anti-A and anti-B antibodies in the plasma. Since antigens are not present in this type of blood, people with blood group can donate blood to people with any blood group type. Besides, people with blood group O can only receive the blood of the same blood group, that is from group O only. That's why group O blood group are known as universal donor, whereas the group AB is known as universal recipient. We'll just brush up that in blood group A we have an antigen of A and antibody of anti-B. Whereas in group B, B antigen and anti-A antibody. In group AB, we have both A and B antigen, whereas there is a nil antibodies. In case of group O, there is no antigen, but it consists of anti-A and anti-B antibodies. And this is a data of distribution of blood group in worldwide. When we see in India, the blood group O is more common and the rare is AB blood group. Importance of ABO blood group. Why it is important in knowing? Medically, it is important during blood transfusion and in tissue transplant. Socially, one should know his blood group so that they could become a member in the blood donor club so that he or she can approach in case of any emergency condition. In general, among the couples, knowledge of blood group helps to prevent the complication due to Rh incompatibility and save the child from the disorder like erythroblastophetalis, which we'll see in detail in the forthcoming slides. Judicially, it is helpful in medico-legal cases to sort out the parental disputes. Inheritance of ABO blood group. We know the blood group of a baby, that is an offspring, comes from inherited from both their mother and father. So gene A and gene B are dominant by themselves and GO is recessive. Let's see some examples like an allele from the mother is A and from father is also A. The genotype of the offspring will be AB. So the blood type Let's see the example of ABO blood group from the inheritance. The allele from the mother is A, allele from the father is A. So the genotype of the offspring will be AA. So the blood type of the offspring is A. And another example is if the allele from the mother is B and from the father is O, the genotype will be BO. So the blood type of an offspring will goes to B blood group. Next, we'll move on to the other blood grouping system, that is RH blood group system. RH blood group system or rhesus system is another most critical system for grouping blood type. Some blood group cells have the presence of RH factor commonly called RhD antigen. The D antigen in the RH system is namely responsible for making our blood either positive or negative. In particular, if we have RhD antigen in our bloods, it is being called RhD positive. Besides, if we lack or there is absence of RhD antigen, blood type is called RhD negative. So, it was first discovered in rhesus monkey. So, hence the name RH factor. There are many RH antigen, but only the D antigen is more antigenic in human. Why we should know RH grouping? RH group system is different from ABO group system because the antigen D does not have a corresponding natural antibody, that is anti-D. However, if RH positive blood is transferred to RH negative person, anti-D is normally developed in that person and that patient comes to the development of antigen against their body. On the other hand, there is no risk of complication if the RH positive person receives RH negative blood because they lack the antigen D. So, how will we classify? If they are RH positive person, the blood group contain RH antigen on RBC surface. Furthermore, they can receive blood from people with RH positive blood and RH negative blood. Whereas RH negative, those who have RH negative blood groups do not contain RH antigen, that is D antigen. Moreover, they can only receive blood from the people with RH negative because if they receive blood from RH positive, that person will start developing an antigen against their own blood group system. Combining ABO and RH blood system. It is because both blood group system together yield a complete blood group type. So, in particular, 8 major blood types are present in ABO-RH blood grouping system. So, like example, if blood group A containing RhD positive, the sign is A positive. And in blood group B, if there is RhD negative, that is B negative. And it goes on like this. Next, we'll see some applied physiology. What are the conditions we could see in case of ABO blood incompatibility or in RH system? First is erythroblastofetalis. Hemolytic disease is the disease in fetus and newborn characterized by abnormal hemolysis of RBC, that is abnormal destruction of RBC. It is due to RH incompatibility, that is difference between the RH blood group of the mother and the baby. So, like already we saw, when the blood is being transfused from RH positive person to RH negative person, that is in first transfusion there won't be any complication, but the RH negative person tend to develop RH antibody in their body. So while in case of second transfusion, RH antigen reacts with the RH antibody. So when the antigen reacts with the antibody, we already saw there is a process which occurs, which is namely agglutination, that is clumping of RBC, which may result in hemolysis of RBC. And some other complications of mismatched blood transfusion. They are hemolysis, which may lead to jaundice, cardiac shock, and renal shutdown. So we have come to an end. We'll quickly recall what all the things we saw in this video. First, we saw the blood grouping system. That is, based on the presence or absence of antigen A and antigen B, blood is divided into 4 groups. They are A group, B group, AB group and O group. In RH blood group, and depending upon the presence or absence of an antigen, namely D antigen, they have been again classified into RH positive and RH negative. Based on this, there are 8 types of blood groups in the human being. And some applied physiology conditions like erythroblastofetalis, which could be seen in an a mismatched blood transfusion from a RH positive person to an RH negative person, and some conditions like jaundice, cardiac shock, renal shutdown, and so on. I hope you all enjoyed this video and got some knowledge about blood grouping system. Thank you.